a very hard science, downfield sort of issue. And I'm just starting to learn how to actually communicate. Right, this with the science arrows. Oh, I see. Okay. So an outline today. All right. So the outline for today, I'm going to talk about some crop breeding technology, which is kind of the starting point for how weeds are becoming herbicide resistant. I'll talk about how that crop technology is regulated and some issues related to crop-to-weed gene flow. And basically what this is the transfer of a gene from a crop to weeds through pollen. And then we'll get to the actual communication part of this that is most interesting for me, is talking about these issues with people both in and out of the scientific community over a large area of the United States. So here is a slide that my good friend Carl Harrell put together. Carl is the person who curates and manages and co-curates and manages biofortified.org. And he likes to talk about crop modification techniques with the general public. And a plant breeder such as myself, somebody who's interested in improving crop varieties for public use, has various tools that we have in our toolkit to work with. So we can do crossbreeding, where you just cross-pollinate different plants with different traits. We have mutagenesis, where you can use radioactivity or even chemicals to create small mutations, and then you can develop a trait based on that. You can use polyploidy, such as in the case of seedless watermelons, where you actually change the number of chromosomes within the cells. You can use protoplast fusion, where you actually merge a couple of cells or cell contents from different species. But for the purposes of this talk, I'd like to focus on these two things right here. There's transgenesis, wherein you actually directly modify the DNA to create a GMO. Raise your hand if you've heard of a GMO. Right? We've all heard of this. GMOs are typically made using bacterial sequences to help introduce the new gene into the plant that you're trying to modify. And that's an important distinction because now, this is a very new technology. We can use genome editing. We can basically make the same DNA changes, but we don't need those bacterial sequences. This is a big deal from a regulatory perspective. Previously in crop breeding, if something was a GMO, because it had bacterial sequences, it was regulated. It was treated like a regulated substance by the government. That means there's all this extra paperwork and money and tests and stuff that have to be done before you can release something to the public. But if something was not a GMO, it had no bacterial sequences, then you just treat it like some normal plant and it can be released to the public much quicker and much cheaper. But now, if we can make the same DNA changes, just minus the bacterial sequences, what happens with the regulatory landscape? And how does that impact technology moving to the public to improve food safety and food health and food nutrition? Big issues that impact the whole world. So a GMO is a genetically modified organism. In agriculture, most GMO traits focus on insect resistance or herbicide resistance. They're designed to make producing the crop cheaper and more effective for the farmer so that you can get more yield. Generally, there's scientific consensus that they're safe. However, a number of public concerns persist, and I'm not going to touch on that debate here. But I do want to provide you just a little bit more information about GMOs that you might not know already. So again, they're treated like a regulated substance by the USDA. That's the regulatory body for plant biotechnology and animal biotechnology in the United States. If you are Monsanto or Pioneer, it takes you about $80 to $150 million per new corn or soybean hybrid just to get it to the market, and it takes an extra 5 years on average to be able to get through the regulatory process. Now, if you are not GMO, well, to give you an estimate, about 25% of that cost is just in regulation. So it's a significant amount of money. Now, USDA APHIS is faced with the position of which biotech traits are we going to regulate now that we can directly modify DNA without bacterial sequences. So this is kind of up in the air. And that's a big deal for commodity groups because, and what I mean by a commodity group is, there's stakeholders involved with different crops. So for the corn and soybean commodity groups, there's so much corn and soybeans grown in the world that they can usually afford the cost of regulation and still turn a profit. So they can invest and still get a return. It's okay. But I work in sorghum, and sorghum is kind of a relative of corn. It's less common in the United States. There's not the same money. And so if we have to be regulated for every trait that we release, I mean, that's going to destroy sorghum. And that's a big issue because under massive international development as a biofuel. We're talking millions and millions of dollars in investment just in the United States. And that's mostly because it has better drought tolerance than corn. How many of you have heard of corn ethanol? Okay. So most researchers agree, even corn researchers agree, corn is not the best choice for ethanol, but it's the best system that we have understood at this point. We're trying in the United States very hard to move sorghum, which is this crop here, into ethanol development. However, the problem is sorghum is a much more recent crop in the United States, and so it doesn't have herbicide resistance traits the way the corn does. And if it doesn't have herbicide resistance traits, then it's a lot harder to control weeds, it's a lot harder to produce a crop and you can still get an economic benefit out of it. And if you notice these plants here, that shatter cane, there's a special case with sorghum, and that is that it has related weed species. So in other words, shatter cane is the same species as sorghum, it's just a de-domesticated variety of it. And so actually, if you put herbicide resistance in sorghum, and now we have that trait, it's called ensign sorghum, it's resistant to some herbicides, those genes can move through pollen to the shatter cane, or Johnson grass is another relative, and make them resistant as well. Well, what happens when your weeds become resistant to the same herbicide that the crop is resistant to? They just spread like crazy, right? And so here our companies spend a ton of money. This particular trait is not Gmos, so they haven't spent, the $150 million typical, but they've still spent a lot of time and a lot of effort to make this crop, to make it useful to biofuels, to the U.S. economy. But if all the weeds become resistant, there goes that investment. So that's a big issue. And then also, is this going to be regulated? So my group at the University of Nebraska, Lincoln, has done some modeling work. And what we've determined is we have some graphs here. So on the x-axis you have year going from 0 to 20 after commercialization of infant sorghum. And then you have plant population density here and on that axis. And then this is the frequency of the resistance gene that came from infant sorghum on this axis. The red line is the frequency of the resistance gene in the population. And the dotted line is the frequency of seeds in the seed bank, of shatter cane in particular. And then the solid line is the frequency of shatter cane individuals that are resistant that are actually growing in the field. And what you can tell is that if you just grow infant sorghum, the herbicide-resistant kind, if you just grow it year after year after year, within about 5 years or less, all of your shatter cane plants are going to be resistant, and the populations are going to be quite high in your fields, and so it's just going to be a mess. So clearly something else needs to be done by farmers to handle this. And what they determined, or what our simulation models have determined, is that if you rotate between infant and something else, in this case you're rotating with normal sorghum, but in this case you're rotating with soybeans, what you see is that the plant population densities in the field, they might be resistant, but their population densities are low enough that they're not producing an economic impact necessarily. You can still recover a crop off of that. So this would be an example of a way to manage herbicide-resistant weeds. And these following slides are just kind of similar scenarios. This is if you're only growing infant once every 3 years, once every 4 years, or once every 5 years. You can see that it takes longer for the populations to become resistant, and the populations of the shatter cane plants remain pretty low, and they don't damage yield too much. So the key here is rotation, rotation, rotation. Okay, so we have this simulation data, but what we need is empirical data to validate that this is actually going to be the case in real farmers' fields. That's really what the USDA needs to know, and that's really what companies need to know if they're going to invest in a new trait. How are we going to be able to maintain weeds at a level that they're not going to provide economic damage? So what my lab did, and this here is a picture of my grad student, we've developed some genetic methods that I won't go into too much, but basically we've developed good fingerprinting of ensign sorghum and shatter cane and Johnson grass, the wild species. And so if a farmer, for example, called us up and they said, hey, we've got a herbicide-resistant weed in our field, is this because of gene flow or what's it from? We could actually genetically fingerprint that and we can tell you this was gene flow or this was not gene flow. So now what I need to do is sell my ideas about monitoring populations across the United States, particularly the Great Plains, of herbicide-resistant weeds with farmers, with university people, with a whole bunch of industry people, people who are just involved in this whole process. I need to get us all on board so we can start to understand how herbicide resistance is evolving in these weed populations across the United States under different cropping systems, so that we can validate these models and give good data to APHIS and to industry. So here's the rationale, the threefold rationale we have. Environmental impacts documented from infant commercialization can inform responsible deployment of future nuclear traits in sorghum. So GMOs, gene editing techniques that affects nuclear DNA. So any traits that we might want to introduce in the future can benefit from this sort of information. We understand the rate of gene flow into the weeds, for example. These traits, such as herbicide resistance, are really needed in sorghum to make it more competitive with maize, otherwise known as corn, and reduce the inputs like fertilizer and water needed for grain and biofuels production. Huge for biofuels industry and for sustainability of the fuels and energy sectors. Identifying best management practices to delay resistance evolution and minimize population densities of resistant weeds will prolong the lifespan of these new technologies when they emerge. So that's kind of the sell. And so far I've been very fortunate. I've been able to work with top APHIS leaders and tell them what I'm doing and get them interested. So the next step, of course, is to get funding and to talk with farmers and the extension people. So that's what we're going to be talking about now. So I've been going and talking with extension people who are outreach people in agriculture, and I've been able to meet with a number of farmers in Nebraska and Kansas over the past several months. And I've been telling them about what I'm doing and telling them that I'm interested in the near future in disseminating some surveys to ask them about how they're managing their crops, if they're interested in including herbicide-resistant sorghum, and if they'd be willing to participate in some kind of regional monitoring program. And in addition, I'm trying to educate them about the special challenge of when you have crop-to-weed gene flow, in addition to just the selective pressure of constantly applying herbicides. So as you can imagine, the weeds can become resistant either through herbicide or through just always applying the same herbicides. And we want to track both of that. And I also want to advertise to them that we are trying to do no-cost population sampling. So that's why we're applying for funds. Okay, so the grower surveys would be followed up after 3, 5, 7, and 10 years to track agroecosystem choice dynamics. That refers to how frequently people are rotating their crops and which herbicides they're using. And we're trying to identify populations of interest so we can use them in detailed studies to really understand the frequency of gene flow versus the herbicide selective pressure version of herbicide resistance evolution. So we have a number of stakeholders I've identified, and in the end we hope to generate some data that we can provide to USDA, APHIS, industry, and commodity groups to reference when they release new nuclear technologies, either through GMO modification or through genome editing modification. And hopefully then we're only regulating the things that need to be regulated and decreasing the regulatory burden in sorghum so that we can have a sustainable commodity group. So that's some of the interesting stuff we're working on, and we're pretty excited to be able to develop these molecular markers to make these screenings possible. These are our funding sources. This is a sorghum commodity group, and DuPont Pioneer is the one releasing instant sorghum. So thank you very much.
We have time for one quick question while we get ready for the next speaker.
Did someone else raise their hand? I was just wondering if you, in terms of sort of the science communication aspect of things, if you encountered any resistance from stakeholders or farmers in participating in this, and what you used to maybe overcome that, or what techniques worked?
Yeah, the biggest problem is that farmers are constantly being surveyed. You know, how do you do this? How do you do that? Everybody wants to study farmers, right? But farmers are like the most busy people on the planet. You know, so how do you actually get them to say, yeah, you can come into my farm and do sampling my weeds, you know, and is that going to impact their ability to get crop insurance if they haven't been rotated properly, for example, rotating their chemistry. So what has been suggested, and this is the idea that I'm following up on, is distributing the surveys at extension meetings. So farmers typically come to extension meetings to learn about new crops that might be available to them, new herbicides that might be available. And so if you distribute the survey right there, then it's easy for them to just kind of fill it out and be done with it, rather than having to mail it to them or, you know, call them in the middle of their daughter's birthday party, or you don't want to pester them. That's